Hello students. So today let us look at locomotion in amoeba. Or this is called as pseudopod movement. Locomotion in amoeba. Although we are writing this as locomotion in amoeba, any cell which has got this kind of movement on a surface spreading, this also has got a similar type of pseudopod movement. For example, even macrophages, whenever they try to move out of their region, spread out and attack other cells, this is the way in which they move. So we call this as pseudopod movement. So let's just draw briefly a structure of an amoeba. That will be its nucleus. So what I am trying to show is that its entire cytoplasm is made of, or can be recognized as, two parts. One is an internal part which we call as endoplasm, and here is an outer part which is called as ectoplasm. So cytoplasm is made up of ectoplasm and endoplasm. This ectoplasm is a little more thicker, so we call it as gel-like. So it's a little more solid. Whereas the endoplasm is more like a liquid. So we call it as a sol. This is a gel. Now, why is it a gel and why is it a sol? So, cells have got cytoskeleton, is made of the cytoskeleton is made up of, and one element is called as actin. Actin filaments, which is a protein. So these filaments, they can make up a network. So if actin is individual, then it is sol-like. If actin is in the form of a network, then it is more gel-like. So if the actin forms a network, so if these kinds of networks are formed, then it is gel-like. Here there is no network, so this is sol-like. Here this is gel-like. So this change of form of the cytoskeleton of actin is responsible for converting sol into gel and gel into sol. And that is what is responsible for ectoplasm and endoplasm. So ectoplasm is more gel-like and endoplasm is more sol-like. Now just before a pseudopodium is formed and the amoeba needs to move, let us say that the direction of movement is in this. So very quickly, the same plasma membrane at this anterior end, wherever it is supposed to move along that direction, it quickly develops a layer called as plasma lemma, and at this anterior end it forms a transparent hyaline cap. At the anterior end, a hyaline cap is formed. Now there is a lot of pressure from all these sides for the amoeba to move along this direction. So from all the ends there is pressure for the amoeba to move and to get pushed along this direction. So it gets pushed. So internally here there is a tube-like region which is getting developed. A thin tube-like structure is developed. And as this thin tube-like structure is developed, that means at this posterior end, of at the posterior end of the pseudopodium, actin network is broken. Actin network is broken Therefore, formation of sol. Okay, and this formation of sol is therefore called as solation. So sol is squeezed anteriorly. So this layer is squeezed along this direction. And as it comes over here in the front, it moves along all the directions, and therefore this part is called as fountain zone. At anterior end, there is a fountain zone where the sol spreads out and pushes hyaline cap and pseudopodium forward. Now when this part immediately at anterior end, sol is converted to gel. The sol is converted to gel. Why? Because network of actin is formed. Why is the network formed? Because there are some enzymes that allow this to function and allow that to take place. So there are certain conditions of when this is individual and the condition gets quickly changed at the anterior end. That condition is altered and therefore the network of actin is formed. That means it is getting converted to sol. To gel. So this is called as gelation. Gelation means it becomes hard. So in the posterior region over here, what is the step around this part? Around this part, the gel is converted to sol. So it is called as solation. And at this anterior end, what is happening? The sol is converted to gel. That means around this here, on this side, this side, this side, all these sides. So this is called as gelation. And as that happens, now everything is got little more solid, but very quickly from the posterior end there is solation, so again it is getting pushed. So how we can imagine this? We can imagine that there is a tube of liquid surrounded by a gel. There is a tube of liquid surrounded by gel, and this tube is squeezed forward, and this tube is squeezed forward, and this extends the pseudopodium. And this is how it can move ahead. For a long time we didn't know much about this, and now this is kind of, we know it. And the earlier it used to be called as the sol gel theory, and we have tried to explain the molecular mechanism by change of state of actin. Change of state of actin. That means from being individual or separate molecules to becoming converted into the form of a network. So that was why it was called as, earlier it used to be called as sol gel theory. So this is how pseudopodium moves, and the same principle is used for movement of other cells also. For example, even in a human being, that is how macrophages also tend to move, by change of state of these organisms, how they spread on a surface. So with this we come to the end of this topic. Thank you.